What I want to do today is we're going to kind of extend our discussion on the GI system. We're going to talk a little bit about the accessory organs and focus a little bit more off of the stomach, going into the small intestine, the large intestines, the liver, and then, of course, maybe throw in a little pathophysiology for everybody and kind of recapping the ulcers and emesis, the vomiting aspect of problems that can occur. So one of the cool comparisons that I want you to think about and review in your mind is we spent some time talking about the cardiac action potential. In 67A, we talked about the nervous system and muscle action potential. And this picture that you're seeing right here is another kind of potential called the slow wave potential. The slow wave potential looks like this. It almost looks like little waves going up and down, up and down. And essentially what you have is you have a very consistent signal pattern inside the enteric smooth muscle of the GI system. And it still works the same way. So this is nothing brand new. You still have a threshold. You still have action potentials. You still have the depolarization and the repolarization. All that stuff is the same. But what you have is you have more of this rhythmic wave-like motion in the slow wave potential. And this relates to the ability for the peristalsis and the segmentation to take place. And this is particularly in the smooth muscle because of that layering. Remember, the GI system has the 4 layers. You have the mucosa layer, submucosal layer, muscularis layer, and the eventia. And so when you think about the rhythm of signaling, these are all localized signals. They're all controlled by the autonomic nervous system, enteric nervous system. And they have that same signal pattern that we talked about throughout the whole body. So the first organ or accessory organ I want to focus on is the pancreas. This is something that isn't too unfamiliar for everybody. This is more of a recap of the exocrine and endocrine functions. So you will need to know, again, the glucagon and the insulin and the somatostatin functions, the alpha cells, beta cells, delta cells, all that good stuff that you had a chance to look at for our first exam. But the exocrine function here is the ability to produce the pancreatic juice. And the one item I want to focus on is bicarbonate. So you remember what we said on Friday is that when the chyme coming from the stomach into the small intestine, this is super, super acidic. This is this rich, milky, digested protein food bolus that has a lot of acidity in there, low pH. And so the very first part of this small intestine has to be able to endure that. And so your pancreas does a job to provide a mixture of bicarbonate to begin the neutralization process. So over here's a picture showing you the pancreas. A little bit of the anatomy here. The gallbladder sits a little bit skewed, tucked behind it. And then you have the leaf-like gland of the pancreas. We have the main duct over here in the center. We have the accessory pancreatic duct that joins down. And then we have the sphincter of Oddi. This is the opening that goes into your duodenum. And it connects down from the common bile duct. So you have a mix. There's an intersection where the juices from the bile and the gallbladder and liver comes in mixed with the enzymatic function of these pancreatic cells. And it is interesting here because this does regulate most of the proteases, the amylases, lipase, all the enzymes your body needs to break down all the food products. So this is a picture that I want to zoom into that is another extension of what we've said before from last week's video. So this is a picture showing you the transport duct inside the pancreatic cells. And you also have, again, on the left-hand side, the capillary blood flow. This is basolateral membrane. On the right-hand side, you have the lumen of the duct. So you're trying to get it across. And so the important feature about these cells and the pancreas is that you also have the carbonic anhydrase enzyme. So this is probably the third, fourth time we've already used this as an example where the CO2 gets modified to carbonic acid. You make protons, you make bicarbonate, and now on the right-hand side, on the lumen side, you have a dedicated chloride and bicarbonate antiport, right? So the chloride comes into the cell for expunging out the bicarbonate. And then you also have this negative ion transporter, and this is what they call CFTR. These are just generic anionic transporters for chloride, other anions to go through, so the chloride can go in and it resets again. So you have this cycling effect of the bicarbonate going into the lumen. So you get the bicarbonate in to now neutralize the acid. On the left-hand side, let's not forget, the proton is still there too. So you have, again, the sodium proton antiport, right? This is something that's not brand new. We've talked about this before. You also have the NAK pump doing its job to maintain that sodium output, so it maintains a gradient. And then you also have the potassium ion channel to allow potassium to go back out. So All of those channels that we talked about over and over are still in play. The only thing here is that you have the CFTR, chloride anion channel, and you also have a chloride bicarbonate antiporter that moves the bicarbonate to one end. All right, so this isn't, again, anything new, but a lot of the themes in the kidneys and inside the GI system and inside the lungs are similar because your bicarbonate is used from head to toe. It's used throughout all these systems. The pancreas also has the ability to generate a series of inactivated enzymes. Some of these enzymes are a little bit more specific. So one particular example is one that breaks down the proteins, the generic bigger chain proteins called trypsinogen. And then trypsinogen becomes trypsin. And this will then in turn activate another one called chymotrypsinogen to become trypsin. So there's a chain of what they call the inactivated to activated enzymes. And then as they get turned on, the pancreas can release them into the lumen to help break down the food products that you want to eat. So all those happen inside the pancreatic juices. This is a nice little list to have some of the names in place. So you might come across the word trypsin, chymotrypsin, elastase, carboxypeptidases, amylases for carbohydrates, lipases are for lipids, and of course, one thing we won't talk too much about are the RNA and DNA found in everything you eat. So anything you eat has an RNA and DNA. And so the ribonucleases, deoxyribonucleases are obviously going to be there. But I want you to be aware of all these proteases that break down the proteins. Okay, so these are all proteases that break down different sizes of the amino acid chain, branch chain to free chain, or from amino acids to peptides, and peptides to the amine group and the carboxylic acid group. So they just strip them down smaller. And as long as you recognize these terms, you should be good. So know the enzyme name, know the substrate, and know the brief action that it does. So this is another feedback system that I want to use to describe how the story gets connected, right? So just to make it very clear for everybody, right? We've said that you eat some food and now your stomach begins to respond by releasing acid. Acid gets generated because of all the chemicals like acetylcholine, histamine, gastrin generating onto the receptors. That causes the proton pump to move the protons in, the chloride follows, and now you have a ton of HCl. HCl makes the stomach region a really low pH of about 2. And now your small intestine has to respond because this acidic corrosive substance is going to burn through your small intestine to respond by releasing secretin. Secretin increases in the blood plasm, eventually binds to receptors in the pancreas, and eventually the pancreas releases the bicarbonate juices that I just showed you. And now the bicarbonate will then hopefully neutralize this pH to go up a little bit. Okay, so this is a flow chart showing you hopefully a clear or maybe more clear vision of what I was trying to say, start from last Friday to today. And then as you progress further down to the intestines, what happens is your small intestine also now has a lot more lipids. Even though we have discussed that there are some lingual lipase found in your salivary glands, there's maybe a little bit of breakdown of fats in your stomach, but most of the fat breakdown happens in your small intestine. And this increase in the fatty acid, and in particularly some of the amino acids, is going to cause your small intestine to increase a hormone called CCK. Right? We had a chart on Friday that showed you what these are going to do. Cholecystokinin is going to increase in the plasma level, and it's going to bind to your pancreas and cause enzymes, exocrine functions, to be released. Now you have a flow mixture of the amylases, the trypsinogen, chymotrypsinogen, carboxypeptidases, and they all come into the actual small intestine to break down the proteins and fats. All right, so that's how the chain begins with CCK. And then the liver also plays a very big role. So the liver is a structure that is going to be in one of the accessory roles, but it does have a dominant ability to generate 5 to 25% of the bile. And what happens is it stores not only all the important minerals and nutrients, but it also gets a first filtration function of the nutrients coming back from the GI system. It does the job to rearrange your blood cells and generate the hemoglobin into bilirubin. And then the last part is also, remember, recapping to the cardiovascular system, is the ability to generate all those osmotic proteins, albumin, the clotting proteins, angiotensinogen, and of course the serobinding proteins that carry the hormones around. So a little anatomy of this is when you go into the hepatic sinusoid or the liver cells, they're arranged in these hexagons. These hexagons have all the central canal. The central vein is going to be this drainage site. On the edges of the actual central vein, you're going to have these bile canaliculi, which actually go in opposite directions. So as the blood flows upwards, the bile, shown here in green, the bile ducts drain downwards, so they go in opposite directions. Zooming into it, you'll see that in a one or closer zoom up of each of these edges, you have what they call the portal triad. Triad means 3. you have one of the bile duct, which goes downwards, and the branch of the vein, and then also branch of the hepatic artery, which flow blood upwards. Okay, and then these cells are able to filter. There's also Cooper cells inside here that you can't see that are resident white blood cells that help you clean off and fight off infections. But this is a very powerful filtration system for the whole body. And then the blood flow and the bile juices go in opposite directions against each other's flow. So when you think about the overarching function of the bile salts, is that you have a small percentage of direct generation of these salts that come from one aspect of the liver, but altogether they combine. They get stored into the gallbladder, and the gallbladder joins into the common bile duct, and that's where you enter into the sphincterology, into the small intestine. A good amount of that eventually gets lost, so then your body needs to be able to have a storage site. And so when people have their gallstone developed or gallbladder removed, then their diet has to change accordingly. So this is an important part of the puzzle. Now, when you think about the overall signaling pathway, or what causes the small intestine to respond most violently, well, the chemical stimuli is going to be fatty acids. So triglycerides, glycerol, lipids, fat-based chemicals found in the duodenum is the one main stimuli to begin to uptake the CCK. And CCK does have receptors in different parts of the accessory organs. So the gallbladder causes contraction, which will then cause a squeezing motion. That's also why when there's some kind of causes, if you have to go back to gallstones, there's some pointer because of the fact that your diet is too fatty based or you eat too much fried foods that can cause your body to overproduce CCK, and therefore over time cause these contractions, which could also disturb what is in there and eventually the gallstones and causing a lot of pain. And then the sphincterology also relaxes. So these two outcomes are how your body responds to CCK in the small intestine. CCK also can bind to your large intestine as well, your colon, and that outcome can be a little bit different. So just be aware this is a chemical that has a widespread function in your GI system, not just only one area. Segmentation is a picture shown here where you actually break the bigger bolus into a smaller chunk, whereas peristalsis is the moving of the food product from one end to the one end. Okay, so just be aware that over time, this rhythmic motion helps you make that food product smaller so enzymes and juices can get in and not decay the bond that breaks up down. All right, and then the last part of the story is going to be your large intestine. So this is your colon. You have the appendix down here, the cecum, and then it goes up the ascending colon, goes across the transverse colon, descending colon, the sigmoid colon, and then the rectum. And again, this is where you have the waste buildup over here, pressure buildup. But this is mostly going to be a water absorption site. And so when you think about the problems to how the GI system gets affected, and I want to go back to Friday's discussion when I said, oh, well, what happens when you have too much acid reflux? Or what happens when you allow these stomach juices to not be neutralized and they pass through to the small intestine? What happens when that happens? Or let's say you decide to take too much antacids and then you neutralize so much of the acidity, what can happen? So one of the outcomes is ulcers. And one of the focus here is that this is pretty common. About 10% of the entire population can get affected. It's a lot more common, but it can happen in the upper GI cavity as well as in the lower GI cavity. And the ulcer is ultimately this eroding damage that leads into the burning and eating effect of the GI wall. And this is oftentimes because of the fact that you have either too much acid or too little production of bicarbonate to neutralize the acid. And so the most common places are going to be regions of your stomach where it could burn through, or the very first part of the duodenum. And as a result of this, you have this kind of constant, almost like an acid pain, burning sensation. The ulcers can get so deep where it starts bleeding, causing you to have some kind of dried, darker brown stains in your stool. And depending on the coloration of the stool, it can indicate the rough location. So when you have brighter red stool, blood in the stool, that could oftentimes be because of the rectum region or colon, whereas if it's a darker brown tint, it indicates from the stomach or the small intestine. And there are some cases where it's very, very severe, where you have it completely perforated. This is meaning that the actual entire membrane is eaten off and there's a hole. And that hole can then penetrate into the peritoneum, which can also cause infections to spread out to other parts of the GI system. So it's very, very bad. Oftentimes, certain drugs like chronic aspirin or using of these can help you reduce the mucin. And so therefore, when you have damaging chemicals of food like alcohol, you're exposing the membrane to more harm and repair. And so the ulcer problem eventually can lead to cancers. And one way that they can assess this is endoscopy, where you can go and take a small camera and endoscope and actually put it through the GI system to be able to visualize areas of burn. And it's a small camera that can go on both ends. So colonoscopy will go from the other end, and endoscopy goes from the oral end.